In February 2025, doctors at Children's Hospital of Philadelphia did something that had never been done before in the history of medicine. They took a 6-month-old baby named KJ, who was born with a fatal genetic disease, and they gave him a drug that was designed for one person on the entire planet, him.
A drug made for one person. How is that even possible?
It is possible because of a technology called CRISPR, and if you have not heard of it, or you have heard the name but you're not sure what it actually does, this episode is for you, because CRISPR is quietly becoming the most important medical technology since antibiotics, and in the last 12 months, 3 breakthroughs have happened that are about to change everything.
Okay, let us start at the beginning. What is CRISPR?
CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. That is a mouthful, but the concept is surprisingly simple. Imagine your DNA is a massive book, 3 billion letters long. Every cell in your body has a copy, and sometimes there is a typo in that book, a single wrong letter in the wrong place. That typo can cause diseases like sickle cell, cystic fibrosis, muscular dystrophy, or the disease that baby KJ had.
So CRISPR is like a spell checker for DNA.
That is actually a great analogy. CRISPR is a molecular tool that scientists borrowed from bacteria. Bacteria have been using this system for billions of years as a defense mechanism against viruses. When a virus attacks a bacterium, the bacterium saves a little piece of the virus's DNA in its own genome, like a mugshot. If that virus ever comes back, the bacterium recognizes it and sends out a protein called Cas9 to find and cut the virus's DNA, destroying it.
So bacteria invented an immune system that works by editing DNA.
Right? And in 2012, 2 scientists named Jennifer Doudna and Emmanuelle Charpentier figured out how to reprogram this system. Instead of targeting virus DNA, they showed you could aim it at any DNA sequence you want. You give it a guide, a short piece of RNA that matches the DNA you want to edit, and Cas9 goes and finds that exact spot in the genome and cuts it. They won the Nobel Prize for this in 2020.
So you can find any gene and cut it, but cutting sounds destructive. How does that help?
Great question. And that is where the story gets really interesting. The first generation of CRISPR was basically molecular scissors, cut the gene, disable it. That works if the problem is a gene doing something bad, like producing a toxic protein. You just snip it and the cell's repair machinery patches it up. But sometimes the repair is sloppy, you get unintended edits, off-target effects, and for a while that was CRISPR's biggest problem. You could edit genes, but you might accidentally edit the wrong ones too.
That sounds dangerous.
It was a real concern, but then came the second generation, base editing. Instead of cutting the DNA strand entirely, scientists developed tools that could change a single letter in the genetic code without making a cut at all, swap an A for a G, fix the typo without tearing the page. Much more precise, much safer.
And now there's a third generation.
There is, and this is breakthrough number 1 from our list. In January 2026, scientists at the University of New South Wales in Sydney, working with St. Jude Children's Research Hospital, published a paper in Nature Communications showing something remarkable. They can now turn genes on and off without touching the DNA at all.
Wait, no cutting, no editing, just turning genes on and off? How?
It is called epigenetic editing. Here's how it works. Your DNA is covered in tiny chemical tags called methyl groups. Think of them like little anchors attached to the surface of your genes. When these anchors are present, the gene is silenced. It is turned off. The cell cannot read it. Scientists have known about these methyl groups for decades, but there was a huge debate. Are these anchors the cause of the gene being off, or are they just a side effect? Like, do they show up because the gene is already quiet?
It is like asking whether the cobwebs caused the abandoned house or the abandoned house caused the cobwebs.
That is literally the metaphor the lead researcher used. Professor Merlin Crossley from UNSW said, quote, "We showed very clearly that if you brush the cobwebs off, the gene comes on, and when we added the methyl groups back, they turned off again. So these compounds are not cobwebs, they are anchors," end quote.
So they settled the debate. The methyl groups are actually controlling the genes.
And more importantly, they showed you can use CRISPR to remove or add these tags on demand. No cutting DNA, no risk of unintended mutations. You're just flipping a switch that is already built into the cell. This could make gene therapy dramatically safer because the biggest risk with traditional CRISPR has always been the cut itself.
That is incredible. So, what about the baby? How does KJ fit into all of this?
KJ's story is breakthrough number 2, and it might be the most emotionally powerful thing that has happened in medicine in years. KJ was born with a condition called CPS1 deficiency, carbamoyl phosphate synthetase 1 deficiency. It is incredibly rare. His body could not process ammonia, which is a natural waste product of protein metabolism. Without treatment, ammonia builds up in the blood and damages the brain. It is usually fatal.
Is there a standard treatment?
The standard treatment is a liver transplant, but KJ was too young. At 6 months old, he was not a candidate. His parents, the Muldoons, were running out of options, and that is when a physician scientist at Children's Hospital of Philadelphia named Rebecca Aarons Nicholas proposed something that had never been attempted: a personalized CRISPR gene editing therapy.
Personalized meaning made specifically for KJ.
Specifically for KJ and only KJ. His particular mutation in the CPS1 gene was unique to him. Nobody else in the world has that exact genetic error. So the team at CHOPP and Penn Medicine designed a CRISPR guide RNA that targeted his specific mutation and corrected it. They manufactured this therapy in record time, and on February 25, 2025, KJ became the first person in history to receive a systemic personalized CRISPR gene editing therapy.
And it worked?
He was discharged from the hospital. As of the latest update in December 2025, he is doing well. His ammonia levels stabilized. He went home with his family.
That is extraordinary. But you said the drug only works for him. Does that mean they would have to make a new drug from scratch for every patient?
Yes. And that is both the promise and the challenge. Right now, designing a personalized CRISPR therapy takes months and costs an enormous amount. But this proof of concept changes the equation. It proves that the approach works. The next step is building systems that can design these therapies faster and cheaper. Kiran Musunuru, one of the researchers involved, said, "This is truly N of one medicine. The drug was made for one patient, this one kid, and it is tuned to his mutation." As the technology matures, you could imagine a future where any child born with a rare genetic disease gets a custom CRISPR fix within weeks.
That would be revolutionary for the thousands of rare diseases that have no treatment.
There are over 7,000 known rare genetic diseases. Most have no cure, many have no treatment at all. Personalized CRISPR could change that equation entirely.
So, what is breakthrough number 3?
Cholesterol. In November 2025, CRISPR Therapeutics published results in the New England Journal of Medicine from their CTX 310 trial. This is an in vivo CRISPR therapy, meaning it is injected directly into the body. No cells removed, no lab processing, just an IV infusion.
What does it target?
A gene called ANGPTL3. This gene controls how your body processes triglycerides and LDL cholesterol. Some people have naturally occurring mutations that turn off ANGPTL3, and those people have extremely low cholesterol and almost never get heart disease. So the idea is simple. What if you could give everyone that same genetic advantage?
One shot to permanently lower your cholesterol instead of taking statins every day for the rest of your life.
That is exactly the idea, and the phase 1 results from 15 patients were remarkable. A single IV infusion produced deep and durable reductions in both LDL cholesterol and triglycerides. No serious adverse events, no significant liver problems. The edits appeared to be permanent.
That could help millions of people. Heart disease is the #1 killer worldwide.
And CRISPR Therapeutics is not stopping there. They have 2 more cardiovascular programs in development. CTX 320 targets the LPA gene, which controls a cholesterol particle called lipoprotein a. High LPA is a genetic risk factor for heart attacks that affects about 20% of the population, and right now there is no approved treatment for it. CTX 340 targets the AGT gene, which is involved in blood pressure regulation.
So, we are talking about a future where a single clinic visit could permanently fix your cholesterol and blood pressure?
With your genes, not drugs that wear off, not pills you have to remember every morning, a permanent change to the source code.
Let me make sure I am tracking all of this. In the span of about a year, CRISPR went from cutting DNA with molecular scissors to rewriting single letters with base editing, to now flipping gene switches without touching DNA at all. A baby got a one-of-a-kind cure, and a single shot permanently lowered cholesterol in a clinical trial.
And there's one more thing worth mentioning. Remember how I said CRISPR's biggest challenge was getting the editing tools into cells efficiently? In September 2025, researchers published a technique where they wrapped the CRISPR machinery in spherical DNA-coated nanoparticles. It tripled the gene editing success rate, improved precision, and dramatically reduced toxicity.
So the delivery problem is getting solved too.
Every bottleneck is falling. The precision is improving, the safety is improving, the delivery is improving, and the first cures are already here. We are not talking about someday anymore. KJ is at home with his family right now because of this technology.
This really does feel like one of those moments where you look back and say that is when everything changed.
Jennifer Doudna, one of the inventors of CRISPR, said she always knew the technology would change biology. She did not expect it to happen this fast, and honestly, neither did most of the scientific community. The pace of breakthroughs in the last year has been staggering.
And we are just getting started.
We really are. If you want to keep following these breakthroughs with us, make sure you're subscribed because the next chapter of this story is going to be even bigger. And if this episode blew your mind, share it with someone who needs to hear it.
Until next time, stay curious.